Welcome back. I'm going to take you guys through a whirlwind introduction to genomic databases, and we'll come back and revisit these in some detail as we go through the semester. Here on the first page, these are links to the main databases we're going to talk about today, so you can link to them by clicking on the name. So if you want to go to OMIM, click on the OMIM link there. So we're going to start back here at our old friend, the National Center for Biotechnology Information. You use this the same way that you searched, at least initially, for PubMed. And you see where it says all databases there at the top. That's a pull down menu, and you could select PubMed from there if you wanted, or any of the many databases here. But you type your search term into that bar, and then you click search. And so I chose to search for this term, phospholipase C gamma, and it returned back results in a bunch of different categories. You can see here literature, health, genomes, genes, proteins. We're going to touch on a few of these today, and then some of the others as we go through the semester, not for this particular example. The first one I want to show you is their books. This is not really at all a genome database, but I thought you might find this interesting and useful. So PubMed, or the National Center for Biotechnology Information, has contracted with some publishers so that it can print the entire text of several books. I think the idea was to keep up with all of the editions, but they haven't really done that. As you can see here, this slide was made today, or yesterday, June 1st, 2015. And if you look at these dates, they range from 2000 for the Madame Curie Bioscience database all the way to 2012 for the Neurological Basis of Suicide book. So you'll see it scattered all over the place. However, I use this database primarily to gain background information on a new topic, so I could look in these books, or to get some nice information and slides for teaching my undergraduate classes. So now if I follow that link that I have circled, the molecular biology of the cell, there's two results in this book, and the top one is shown as signaling through enzyme-linked surface receptors. And underneath that, there's two detailed results, and you can click on one of those or any of those that you want. I clicked on the bottom one, the phosphorylated tyrosines link. That takes you here. And so it gives a really nice description of this topic idea with respect to phospholipase C gamma. So this is pretty useful. You can, of course, get this same information by reading actual papers, but it's a lot more detailed, and sometimes it's nice to get your background information first rather than get the details. I also like the inclusion of figures as shown here. So by the arrow, if you click on that figure, it brings it up large size like this. And there's some nice detail here, and would be a very nice slide for introducing the concept of signaling through phosphotyrosine and the SH2 domain of PLC gamma, and describing how PLC gamma interacts with the platelet-derived growth factor receptor, shown in A there, and then in B it shows you a three-dimensional view of the SH2 domain of PLC gamma and how it might bind to phosphotyrosine, and then a little conceptual drawing there in C about how they interact with each other. So this would be a good, I would say, nice introduction to that topic. So many of you are going to be teachers one day. You may like this website for this very reason. Okay, now back to our databases. The first real database I'm going to take you to is called OMIM, which stands for Online Mendelian Inheritance in Mankind. If you click on that link, here's OMIM. We had searched for phospholipase C gamma, and that would come up here if I'd clicked on the right place. But I first wanted to introduce you to the database. The OMIM database is a big database that is categorizing human genetic phenotypes and genotypes, and where known, actually down to the actual mutation in the gene that has been linked with the phenotype or the disease phenotype. It's a pretty nice database. Also, I use this for teaching, but it's also useful for developing research ideas. Oh, sorry for that delay there. I'll see if I can cut that down a little bit when I save it. So back here, a little bit more about the OMIM database. You can see some of the statistics. It's updated every day. Um, there's right now almost 23,000 different records in the OMIM database. 14,000, almost 15,000 of them are linked to an actual gene description that is linked to a phenotype, which I find pretty amazing. And you can see it's broken down into 14,000 autosomal genes, almost 700 X-linked, 48 Y-linked, and even mitochondrial linkage to um, human genetic diseases. I'll show you some of these records so you can get an idea of what they contain. It's lots of stuff. The morbid map, in this case, morbidity refers to the development of a disease, not necessarily just death, but so developing cystic fibrosis or developing diabetes would be a morbid phenotype. Most of them, the morbid phenotypes, are on chromosome 1. Not too surprising, it's our largest chromosome and has the most genes, and the fewest are on the Y chromosome, which is also not surprising. There's only really a few genes on the Y chromosome itself. Now, so back to the OMIM database. You can search it in a variety of different ways. You can link to it from the NCBI all databases search, as we'll see in a moment, but you can also search the database by itself by using keywords. In this case, I searched for a disease, diabetes, and 953 records have some sort of link to diabetes. The one that I'm interested at this point is insulin dependent diabetes mellitus, or what you probably know as type 1 diabetes or juvenile diabetes. This is linked to a chromosome position on chromosome 6. And if we click on that link, it will take us to its page. There's lots of stuff on this page. You can see on the left is a description of the disease, some clinical features. We'll scroll down and look at that a little bit more in just a second. On the right is the table of contents, and each one of these is a link. So if you click on any of those, it will take you to that part of the record. Like, for example, later we'll look at animal models or also clinical trials, which is under clinical synopsis. So as you scroll down, I just cherry picked a couple things you might find interesting. You can read about the clinical features of the disease. Sometimes that links to particular genes or alleles, or sometimes it tells stories about particular families that were instrumental in learning about the diseases. Sometimes that makes for interesting reading. And then often there'll be a section on the molecular genetics or the molecular biology of the disease, so you can learn about the gene, sometimes the mutations, and those kinds of things. It's often also interesting to see if there is an animal model for specific human diseases. So there is one or more models for this type 1 diabetes, and it will tell you about either the discovery of the animal model, which it sometimes happens naturally, or sometimes they have to be created. And in that case, it will tell you about how the model was created and links out to the paper. So all of those blue links there will get you to eventually the PubMed database record for those journal articles. So you can quickly gain or get tons of information. Now, the other thing I thought you might be interested in is that for many of these, uh, there's links to records in the clinical studies database, which tells you whether or not there is a clinical trial that's currently happening or has just completed, either on a particular disorder or involved with a particular gene, that kind of stuff. In this case, there were 1,493 studies for insulin dependent diabetes mellitus. I'm showing you four here. Um, this is a big table. You can scroll down and look at all the studies if you do this yourself. I'm going to look in more detail at the second one that says it's recruiting for imatinib treatment of type 1 diabetes. So if you go to that, it tells you where the sponsor, so much of the research might be at UC San Francisco. Sometimes it's spread out throughout the country. Um, it will tell you the purpose of the study, in many cases how the study is designed, whether or not any results have been found. And if you, I only took a tiny part of the record, but you can scroll down and find out what type of patients they're looking for, who you need to contact if you were interested in becoming a study participant. And even for doctors, sometimes they can enroll their patients in these kinds of trials. So pretty neat, pretty interesting, and often extremely useful. They have this for all the different morbid gene studies in here: diabetes, cystic fibrosis, all types of cancers, etc. And in many cases, they're updated extremely recently. This one was last updated on March 20th, 2015. So it's pretty recent. So back to the database record in OMIM for diabetes. Um, we're back to the table of contents page just to show you a few more things that you can link to from here. Everything in blue here is a link, and so if you click on it, it will take you to that actual um, database. So the first thing I clicked on there was the gene location. This is on chromosome number 6, and they're right down there in middle. In the middle, I've highlighted. It's highlighted in sort of peach color. Is the gene linked to this OMIM record? All the other genes, like the one just above it, the inflammatory bowel disease number 3, or the major histocompatibility complex, etc. These are all genes, neighboring genes on the same chromosome and pretty much right there in the same location. Not the same actual DNA sequence, but these genes are all located right next to each other. Sort of, if you think of this as a neighborhood on a chromosome, these are all closely associated or closely uh, built houses, one after the other. They're all neighbors. If you follow the link to the genome browser, which you can get to through OMIM, it will actually show you that part of the chromosome. So here we are. This links out from NIH to the UC Santa Cruz genome browser. This was developed as a different resource, but it's such a very good resource that the NIH has incorporated it into its own database by providing all kinds of links to it. This shows you the chromosome location where we were on chromosome 6. We're spanning about 6 million base pairs in the record being shown here. If you look towards the top, it shows you a picture of chromosome 6 with a little red box, and we are in that area of the red box. Now, this is a really far zoomed out view, so there's not a whole lot of you right now. You can't pull from this tons of useful information, um, but every single line here has something informative. So as you go down from the top to the bottom, in the bigger box, it says, one of them says ref seq genes. We're going to go look at the database record for that in a little while. But all those vertical lines show you where a gene is located. And so there's lots and lots of genes in this one 6 million base pair area of this chromosome 6. Uh, just underneath it are human messenger RNAs, and then underneath that are the spliced express sequence tags. And so if you zoom in on all of these, if you're knowledgeable about that or interested in that area, you can get quite a bit of information about this part of the chromosome. You can also zoom around the chromosome using the buttons on the top. So the ones that say move, you can zoom to the left, zoom to the right, just like I sort of, an analogy here would be using Google Maps. And if you zoom into Street View. So we're not quite at Street View here yet. We're zoomed up at like the neighborhood level. But if you zoomed in, you could actually get all the way down to looking at the individual nucleotide bases for every one of those genes. And you'll be doing that in other exercises. As we go through the record here, I zoomed in a little bit. So if you notice the picture of the chromosome at the top now, instead of having a little red box, it's a red line. And instead of covering 6 million base pairs, we're now covering 50,000 or 51,000 base pairs. And you may not see where that information is, but it's all up there at the top where it says position and search. So we're getting closer into where that actual gene was located that we saw initially in the OMIM database. Now, the bottom part of this record you may or may not find interesting, but those are called SNPs, which are single nucleotide polymorphisms. So every single one of those is a link to a record from a real live individual human being where they sequenced that gene for that human, and they found a polymorphism at that location. And so these can all be lumped together. Obviously, at any one position, you could have one of 4 nucleotides, an A, a G, a C, or a T. But in most cases, most people have only one of those nucleotides. And so if the nucleotide changes, that's a mutation. Sometimes that makes a difference, sometimes it doesn't. That's all recorded in here. Well, here I clicked on one of those links to one of the SNPs. I chose it randomly, and so it looks like this is categorized as SNP number RS69077002, and so that is from human DNA. And at that position, in most people, there's a C, but then in the SNP shown here, it looks like observed in humans is a T. And so at that position in that gene, there could either be a C or a T at that position. And so that is a polymorphism, more than one morph or more than one nucleotide at that position. Now I know this doesn't make tons of sense right now, but this is pretty interesting because as they categorize these SNPs in humans, this is going to eventually make its way into your doctor's clinics. And sometimes some of these SNPs, whether you have a C or whether you have a T, makes a big difference about how you might be treated for a certain disease or what kind of drugs you can take. The overwhelming majority of the time, whether you have a C or a T, doesn't matter at all, but sometimes it does. Okay, back to our lecture. We're back to OMIM, and all the way back to the beginning where I searched for phospholipase C gamma in the all databases search. And now I've come back here, and this is the OMIM database record for PLC gamma. So this is a different way to search it. Rather than searching for a disease, we're searching for a protein. And here you can look and see what kind of changes in this protein could actually be linked to a human genetic disease. So you'd have to go down through the table of contents, expand all of that, and look to see what's known. And so that's sort of just a different way to do it. One nice thing about this is you can learn a whole lot about a new protein or a new gene in the OMIM database because you can link out to all these different papers. And so here, these are links to all the PubMed records for these these 6 publications shown here from the PLC Gamma OMIM entry. I linked out to one of the papers. I know you're familiar with finding papers. And so this actually is the paper where they found in 1988 the sequence similarity of PLC with a region of the Sark tyrosine kinase. And that's how the SH2 domain got its name. The first, there's an SH1 domain. It's not found in PLC gamma, but it's found in Sark. And so other proteins were found to have this region of similarity with Sark, and so they called that the Sark homology domain 1, or SH1, which is actually the kinase domain. The SH2 domain is a phosphotyrosine binding domain. It was found in Sark first, then in PLC gamma, and now in about 17 or 18 other proteins. And so every protein that has the Sark homology 2 domain at some point in its life cycle will bind to phosphorylated tyrosines. And so you can find this kind of information through the OMIM database. Now we've zoomed back out, we've gone back to the OMIM gene map and looked at the location. So the PLC gamma 1 gene in humans is found on chromosome 20 on the Q arm between 12 and 13.1. The gene symbol is PLCG1 or PLC1. If you click on that location there, it takes us back to the genome browser. And there you can see a picture of chromosome 20 at the top. The red line shows us the location of the phospholipase C gamma gene. And then underneath it here, we've zoomed in on the gene itself. And so we're only covering, in this case, 38,197 base pairs. But if you go down to the next line under the picture of the chromosome, you can see the actual PLCG1 gene map. And there's vertical lines and then horizontal lines with little dashes. So that shows you the direction that the gene is going on the DNA. So it's going from left to right. But all those little vertical lines are exons. And so to make the messenger RNA from that gene, you have to do quite a bit of splicing between all those different genes. All those different exons. Splice them together into an RNA. Okay, excuse me for a second. I was moving some stuff around there. So back to the recording. Um, you can see lots of other things here. If you go down towards the middle bottom of this, it shows you the comparison between the human and genes of other animals. So that's there, like the rhesus monkey, mouse, dog, elephant, possum, all the way to zebrafish. So this gene is found in a bunch of different kinds of animals. It also shows you common nucleotide polymorphisms that are found. You can look at those in the databases. You can also, from this chromosome, you can zoom in to see nucleotides, or in this case, I've zoomed all the way out to see the entire chromosome. This is not that informative actually because you're so zoomed out you can't really see anything, but this should show you all the different genes that are located on this chromosome. There's quite a few, and quite a few that make RNAs and a bunch of different SNPs. Interestingly enough, here it will show you that over chromosome 20, there's quite a bit of homology to other animals, even possums, chickens, zebrafish, the frog. So we share a whole lot of sequence homology with these different animals. And here I have now, rather than zooming out, I zoomed all the way into the base, nucleotide base. You can see the chromosome there. You can see the individual nucleotides of the gene. Rather than having 65 million base pairs across the screen, we're looking at 113. So we're looking at a small part of the PLC gamma gene. Actually, it looks like we are within an intron and not actually an exon of the gene, although there is a part of a human messenger RNA there for one of the records. And so you can see that if you spend enough time looking at this, you can get quite a bit of information, particularly for genetics, which can be quite useful. Okay, back to the OMIM database on NCBI, and let's look at a few more things. So this tells you about how PLC gamma was initially cloned, how it was found. You can link from there to the gene database at NCBI, and so now we're no longer in the OMIM database, but we're in the gene database to read about human PLC gamma. The main thing is over here on the right. You can see all the different things you can link out to. So you can read about the gene, you can see the sequence of the gene, you can look for mutated known mutations of the gene, you can get more information about how it's located on the chromosome. Often you can tell how the promoter region of the gene, how it's expressed. There's also resources for scientists. If you look on the right and the bottom, you can order a cDNA clone, you can go find antibodies, you can look for conserved domains and express sequence tags, all of these that would be useful for you in the laboratory. We'll explore some of this, but we certainly won't have time to explore all of it. I would quickly lose your attention. So here we've clicked into the examine more about chromosome 20, and this is on the NCBI website. This is similar to the UCSC browser, but it's a little bit nicer formatted. So on the top you see chromosome 20 showing you the location of a variety of genes. The PLC gamma 1 is in the middle there in red. Genes are going in both directions on this chromosome, as you can see. There may be a tiny little bit of overlap between the genes, but it doesn't look too much. Then what I'm pointing out here is if you come down to the bottom, you can see the exons that would need to be spliced together to make the RNA. It shows you the location on the chromosome. But I'm going to click on the link that says FASTA. And that takes us here. So this says human chromosome number 20 between what is nucleotides 39,766,161 to 39,800,000. So this is the area that contains the PLC gamma gene, and you can actually see the entire human genome sequence for that area. So the PLC gamma gene is hidden somewhere in there. Then we zoom back out and go back to the sequence viewer. This shows us the different messenger RNAs. Those numbers in blue there where I have the arrow pointing, it says messenger RNA, those are accession numbers. And those are links, so you can actually go to the database record for the RNAs. So here I clicked on one of those. This took me to the FASTA sequence for the messenger RNA for PLC gamma 1. So now this would be part of that chromosome 20 would match to this, but it wouldn't match 100% because to get a messenger RNA, you express from the gene the entire RNA sequence, then you splice out all the introns and splice together the appropriate exons to make the RNA. Here's other parts of it. Showing you just the database record with the FASTA format shows you the nucleotide sequence, but these database records are pretty complex, and so you can get tons of information from them. So here we're looking at the database record for the RNA. It tells us the genes are the papers where this information has been published. So you can go back often and find the initial paper. It tells you more information here. A lot of this is just organizational and categorizing it. So like it tells you how many base pairs the mRNA has, people that cloned it, those kinds of things. If you scroll down the record, it will each often have a link or information from the protein that's made by this RNA. So there is the amino acid sequence of the PLC gamma 1 protein from the PLC gamma 1 RNA. It also links out to several other databases. A lot of these have overlapping information, but sometimes they're different. The variety of different things you see here, like on the top where it says EC number 3.1.4.11, that is the enzyme number, and that's more of a chemist's way of categorizing it. And so every single molecule, a biological molecule, and other molecules, chemicals, are categorized by an EC number, which helps you keep track of them. This is now back to the sequence information for the genome. If you want to understand the relationship between the RNA that codes for the protein and the genome sequence, it's difficult to do that just by eye. And so we're coming back now to the UCSC Genome Browser website. We're going to run a program called Blat, B-L-A-T. In this case, I put the mRNA sequence for PLC gamma 1 into this Blat search query. There are a lot of pull down menus here, but we're going to leave everything alone and just use the whatever is provided standard here. Blat is similar to a program called Blast, except what it does is it takes the messenger RNA sequence and it matches it to the genome, and it splits it up so that you can see where each part of the RNA came from in the genome. That doesn't make sense right now, I know, but I'll show you now. You click on Submit. It found these 5 different results. The one on the top has 100% identity. It's on chromosome 20, which we know this gene is on. And over on the far right, you can see it spans 38,197 nucleotides, which is pretty darn big. So I'm going to click on Browser. You can see I have it circled. This takes us to the genome browser. I just wanted to confirm that yes, this is where we were before. So you see number 1, the chromosome map. You see the location with the red vertical line. And then number 2 points us to the mRNA organization on the genome. Let's go back to our Blat results, and then I'm going to click on the link to details, and that takes me here. Now this is showing us chromosome 20, not the RNA, but the actual chromosome. And notice that the nucleotides are numbered, so there's 50 per line, and some of them are different colors. The different colors there, watch as we scroll down, we start to find here in blue, that's a region of the chromosome 20 that matches almost 100% to the PLC gamma 1 messenger RNA. And this is identified as block 1, or another way to think about it is exon number 1 in uppercase letters, and then in lowercase black letters are the introns. If we keep scrolling down, you see there exon 2, 3, 4, etc. Some of them are really close to each other, some of them are farther away, but whoops, sorry. All of those are exons for the PLC gamma 1 gene. They get spliced together after the RNA is made to make the mature messenger RNA. Now, the Blat is the best tool that I've found for doing this. Okay, now let's talk about one other database. I'm going to focus mostly on the National Center for Biotechnology Information, which is the one I'm most familiar with. But all of this can be done using this other database called Ensembl. Ensembl is virtually the same, but it has a little bit of a more modern interface in some cases. So some people would prefer to use Ensembl. You can see you can do Blast, which we'll talk about later, pretty soon actually. You can do Blat. You can do database searches like we just did using the Ensembl database as well. So in this case, I searched for a different gene. This one's called RAS, human RAS, or HRAS. The H actually stands for Harvey, but it's a RAS is a gene found in it's a proto-oncogene in our genome. When it's mutated, it can lead to cancer formation. It's found in lots of colon cancers. And so here, I searched for RAS, and you'll notice one thing here. We found the HRAS family GTP binding protein messenger RNA, the complete coding sequence. So there's the mRNA there. The database record here is from Ensembl, and I'm showing you the FASTA format. The FASTA format is the simplified format that you can use in programs. It doesn't contain all the extra information, but you can, if you want, get all of the extra information from this database. So I took that sequence for HRAS, I pasted it into the query sequence here for Ensembl. So notice that I'm searching, not using a keyword or anything, but using the sequence. And it searches the Ensembl database, and it finds there on the X chromosome, there's one. On the tip of the chromosome 11, there's a link. And so HRAS is a gene that may have been duplicated. So one thing I like about Ensembl is this particular view. It shows us all of the human chromosomes, and it shows us where we found sequences that matched our sequence. I also sometimes find it useful to search the database using information about the DNA sequence, or not really, not using keywords, but actually using the DNA sequence or the protein sequence. That's on the Ensembl database. It will also list them here in a summary table, so you can see how long the match was, the percentage of identity for the match. And this is sort of like Blat. You'll notice that on chromosome 11 we had 4 matches in a row that were all really close to each other, and there was a 100% match. That makes me think that might be the real one. But you can click on each one of those and look at the matches. So here we are in chromosome 11. You can see it's 100% match. The one on the top is the query, which is the sequence we sent out, and the one on the bottom is the subject, which was identified in the search. Okay, then you can zoom out. This sort of looks like the Blat results here, and find your exons on the genome information here for chromosome 11. So this is similar to the Blat search that we did, but just a little bit of a nicer formatting. Then we can zoom out here, put it on the chromosome. This is like the chromosome map. Our HRAS gene is shown there right in the middle, and bounded by the little red box, and shows you where much of the gene would be located there. This is zooming out a little bit. It shows you the HRAS gene and the different RNAs that have been shown from it. Now, this is a nice example of alternative splicing. So the HRAS, they found all these different processed RNAs, and so the HRAS gene has multiple exons, and they must not all get used, depending upon which HRAS protein has been found. So you can get different HRAS RNAs from a single HRAS gene. Okay, you made it to the end. I hope this didn't take too long and you kept your interest all the way through. Here's your homework for the week. It's due next Monday, so this is Tuesday, so you have almost a week to work it. June 8th, and just answer these 4 questions and submit it through our course website on Canvas. Thanks for your attention, and I will talk to you again soon.